Hello everyone, and welcome to your first video for the year in Pre-AP Physics. Tonight we're going to talk about dimensional analysis, conversion factors, SI units, and a little bit of the scientific method. This whole PowerPoint should serve as just a review to you guys. I'm sure you guys do this every year in science class, but I still need to make sure that we're all on the same page and that we all remember this stuff. So, you will have a quiz over these things on Tuesday after the 3-day weekend. So if you need to come back and review this video, please do so, again, between tonight and Tuesday. And feel free to do the extra problems that I have in the PowerPoint. Those are designed for you guys to do on your own, just to make sure that you understand the concepts that I'm trying to present. So let's go ahead and get started. So the first thing we're going to talk about are significant digits. Now, I'm sure you guys did these a lot in chemistry. I'm not going to be too crazy about significant digits in physics. You do need to know what they are, and you do need to know what they mean when you see something that has a significant digit of 3 or 4. So significant digits are a way to express how confident we feel in a measurement that we've taken. So if I have 3 significant digits in my answer, I know that I am very confident of my result up to that particular decimal place. This becomes important when you start doing labs because you don't want to be reporting more significant digits than what your instrument can realistically measure. So this brings in a little review real quick about accuracy and precision. So accuracy is how close you are to the true value. So if the mass of an apple is a kilogram and you weigh it and your values are very close to a kilogram, then you have very good accuracy with your particular device, probably a scale. Precision is the degree of exactness of your measurement. That's how your textbook describes it. I like to say it's how well you repeated your experiment. So if you did the experiment 3 times and you got relatively close data all 3 times, then you were very precise. It might not be very accurate data, but it was very precise. Alright, so back to the idea of sig figs or sig digs or whatever you guys called them in chemistry with significant digits. Just be very careful in a lab situation like I mentioned before. If you're using a meter stick, this is a completely inaccurate measurement. There's no way you can measure to 5 decimal places using a meter stick. Maybe you get to 2. Maybe 3 if you're really, really lucky. For the purposes of lab, try to keep all of your values to 2 decimal places unless I specify otherwise. If I see numbers like this in a lab, I'm going to count off just because you're being sloppy with your data, and it doesn't really make that much sense. Okay, here's a little graphic to show you the difference between accuracy and precision in case you forgot. Sorry, it looks a little rough there. Ugh. Anyway, so over here we have accurate and precise all together, all in the center. Not accurate but precise, meaning they're all together but they're not in the center. This is, I don't even know what that is. And this is accurate but not precise. So they're all close to the center but they're just not close to one another. You want to try for this, of course, all the time. Alright, scientific notation, of course, powers of 10. Here's a couple little quick examples for you guys to just review scientific notation. If it's still really confusing to you, please come and see me in tutorials. This is going to come up a lot. So this first one, of course, would be 5 times 10 to the negative 3. And this other one would be 1 times 6. 1 times 10 to the 6th for your answers for these two. Okay, conversions and dimensional analysis. You guys probably did this a lot in chemistry, again, with the picket fence method and converting from moles to grams and back again. We do it a little bit in this class, so just want to make sure everyone is cool with that concept. Before we start doing some practice problems with that, here's a little review for SI units and prefixes. You guys are probably familiar with the kilo and the centa and the milli, but you probably didn't use micro or pico or mega at all in your previous science classes. We'll probably be using these a lot more second semester. This first semester they don't come up too terribly much. So we'll have a review of those when we get there if we need to. Okay, here is some conversions practice. This is, of course, going from grams to micrograms. This grams is the SI unit for mass, remember. Meters, or I'm sorry, kilograms is the SI unit for mass. Meters is the SI unit for length. And the SI unit for volume is liters. So remember those. So conversions practice, the first one we can do together, convert 16 grams to micrograms. So pull up my practice sheet here. So micro prefix, it looks like a funky little u, and it means times 10 to the negative 6th. So 16 times 10 to the negative 6th grams, and we're going to go, is what that means. Now you guys probably have done this with the picket fence method, and you cross off your units to make everything jive up. And you can do it that way if you'd like. If you know a different way that works for you, please use it. I'm not going to be too picky on the method, as long as I can follow your method and your end answer is the correct one. So luckily for us, we're just going from micrograms to grams, and we know that micro means 10 to the negative 6th. So it's 16 times 10 to the negative 6th grams. Now if I wanted to put this in proper scientific notation and make it look a little prettier, I could do 1.6, and then I would have to change my exponent to be 10 to the negative 5th grams. So this is our answer for that one. So let's go back to the other slide. Convert 0.75 kilometers to centimeters. Now again, I like to go when I have a situation where I'm going from a very large number, so kilo means 1,000, to a very small number, centa. I like to convert kilometers to meters first and then go from meters to centimeters. That helps my brain a little bit, but if you guys can just do the straight conversion from kilometers to centimeters, then by all means do it. But I'm going to take the long road here. Right? And in 1 kilometer, there are 1,000 meters. And then in 1 meter, there are 100 centimeters. So we would do 0.75 times 1,000, because these guys cancel out, and these guys cancel out, times 100. So we would do 0.75 times 1,000 times 100. And you get 75,000 centimeters. Alright. For the last one, you guys feel free to try that one on your own if you'd like. And we can talk about it tomorrow. Okay, dimensional analysis. Conversions is when you're going from one prefix to another, but you're using the same unit. So you're going from kilometers to meters, or you're going from micrograms to grams. Dimensional analysis is when you don't necessarily use SI units, and the units don't really jive. For example, you might be asked to convert something from days into seconds, or from meters into miles. So you need to have a conversion factor, first of all. And then you're going to use the same kind of picket fence method that I just showed you guys. So let's look at this one. How many seconds are in 4 days? So let's do this one together here. So if you have 4 days. Now, if you try to go just from 1 day to seconds, it might seem a little overwhelming. So we're going to go to the next largest unit of time measurement in a day. So in 1 day, that would be hours, and that would be 24 hours in a day. And then in 1 hour, there's 60 minutes. And in 1 minute, there's 60 seconds. So all of your units cancel out except the seconds, which you want over here at the end. And then you're just going to straight multiply across. 4 times 24 times 60 times 60. And your answer is 345,600 seconds. Alright, this is a second example that you guys can do on your own to convert 2 miles to inches. I've put your conversion factors there that you need in order to do that one. All right, here are some other practice examples for the dimensional analysis. If you guys are really rusty on this, please go ahead and give these a shot. I'd really like everyone to do this first one just because, and this is supposed to be centimeters cubed, just because it has that centimeters cubed unit in it, and that makes things a little bit weird. So give that one a shot. All right, scientific method. Review the scientific method. There are 5 steps. Sometimes 6 depending on which thing you're looking at. The first is your observation, and then, or sometimes it's called the purpose, and then you have your hypothesis, then you have your procedure, data, and then your conclusion. All 5 of these things will always be in your lab reports too, and we're going to talk about lab reports later this week. These are some vocab words from scientific methods, so just to review for you guys, independent variable, we'll do control in a second. Remember, the independent variable is the variable that you are testing. It's the one that the experimenter is controlling. So, if I'm trying to determine how much fertilizer makes plants grow, the amount of fertilizer is going to be my independent variable. I'm deciding how much fertilizer each plant receives. The dependent variable, this is the measurable variable. So it's the outcome. It's what you're trying to determine. So in my previous example, the dependent variable would be how much the plants grew or how tall they grew. A control, this is something kids always struggle with trying to define, but a control is your experiment without the independent variable. That's kind of how I look at it. So my control in my previous example would be how much the plants grew without any fertilizer on them. How much did they just grow normally before I started my experiment? All right, and then the last little thing that we need to discuss is the difference between a hypothesis and a theory. You guys probably learned that a hypothesis is an educated guess. I'm not a big fan of that definition. I feel like when you guess at something, there's nothing really educated about it. A guess you take when you have no idea what's going on, and there hasn't been any observation or data collected beforehand when you make a guess. So a hypothesis, I don't really think it's that. It's more like a testable prediction. So it's something that you've observed in nature, you've observed a phenomenon, and from your previous knowledge and from what you see in front of you, you're going to make a prediction about what would happen in that particular scenario. So that's kind of the difference for me. I prefer this definition for it. But anyways, it's something that hasn't been tested yet, and it can be right or wrong. It's perfectly fine if your hypothesis ends up being wrong. A theory, on the other hand, is a hypothesis that's been tested, and it has been developed to explain something that you've observed in the world. A theory comes from repeated observation and testing. So maybe in just normal conversation, you guys will use these 2 words interchangeably. You might say, I have a theory about that, or my hypothesis is such and such. But in reality, they're 2 really different things. Alright guys, and that is going to end this video for tonight. Please bring your notes tomorrow to class so that we can review the independent practice problems presented in this PowerPoint, and so I can answer any questions for you guys if you have them. Thanks. See you later.